Hi, it's Ms. Bastido here to talk about control of gene expression. So in the last video, we talked about the idea of how do we get from DNA to a protein. And so we talked about transcription and translation. And now we're going to talk a little bit, this is kind of a hard topic, about how are those genes expressed. So we know that we have all of this DNA in every cell in our body, but we don't necessarily need to make every protein in every cell in our body. And so those genes will sometimes be turned on and sometimes be turned off. And there's different levels of control that we do in order for that to happen. So control is a bit different in prokaryotes than eukaryotes. I'm going to focus on eukaryotes in this video. There are, with prokaryotes, we could talk about lac operons or trip operons, but for here, I'm just going to talk about the eukaryotes and the eukaryotic control of gene expression. So again, this is going to be really an overview. This can get actually quite complex, and the science of this can be quite complex. So here are the general ways, and if you can categorize them this way in your head, then that will help you understand them a bit easier. So we have, I would put it into 5 categories here. So we can control gene regulation at the chromosome level. We can control it at the transcription level, post-transcription, during translation, or post-translation. And so these different levels give us different ways to control the amount of protein being produced. And in some cases, completely turns off a gene so that that protein's not produced at all in that cell. So we'll kind of go through each one of these. This is certainly not an exhaustive list, just talking about some general ways in which we do that. And so one way is through chromosome regulation. And so in chromosome regulation, we have, just as a bit of a review, so DNA is this double helix, and it winds up really, really tightly around these histone proteins, around 8 of them actually, and then is secured by a 9th, and we call that a nucleosome. And that nucleosome then winds up with other nucleosomes to create eventually chromatin fiber, which then goes on to even coil even further to get to a chromosome. And so this being coiled up, when the DNA is coiled up so tightly, there is no way that RNA polymerase can access it in order to read the DNA, to make the mRNA, to then make the protein. And so this is one way that it's regulated. There will be parts of chromosomes that will be permanently coiled in certain cells. And so this can be regulated even further by what happens here in the nucleosome. So here are the histone proteins. The purple is the DNA wrapped around these histone proteins. And methyl groups can be added to the histone proteins that essentially cause the packing of this DNA to even be tighter. And so this packing here, essentially, you can think of it, makes the DNA not accessible at all to RNA polymerase, and so it turns the gene off. And if the methyl group is there, this gene will be continually turned off. On the flip side of that, acetyl groups can be added to histone or chromatin, which would then cause it to loosen up. And when it loosens up now, the transcription factors can bind, and then that allows RNA polymerase to bind. And so both methylation and acetylation are turning on and off the genes by coiling up the chromosome. So it's still chromosome regulation. And we call this epigenetics, and we will talk about it a bit later in this video. So then the next level of regulation would be transcriptional regulation. And this was already discussed in the other video when we talked about transcription, but the idea that we have transcription factors which act as activators. And so those transcription factors will bind to the promoter region, and that allows RNA polymerase to bind, which allows the reading of the gene. And so without these transcription factors, and remember that there would be separate genes for these transcription factors. And so if the transcription factor gene is turned off, then this gene would also not be working either, because it needs the transcription factor in order for RNA polymerase to further read that gene. we've talked about this already as well, but in post-transcription, one way for regulation is how we splice genes. And so we make a pre-mRNA, that mRNA is then spliced, and based on the splicing, we would produce different proteins or different polypeptides, which then go on to make proteins. And so we know that we used to think one gene produced one protein. We now know that one gene might produce multiple proteins depending on how the splicing happens. And so this splicing could also be a way of regulating how much of each of these proteins are produced. Remember in the last video we talked about before the mRNA leaves the nucleus, it gets a 5' GTP cap and a poly A tail. And that is important for regulation of the gene because we can regulate the amount of protein being produced. So the longer this poly A tail here, the longer it will last in the cytoplasm. So there are these ribonucleases that essentially digest mRNA that's in the cytoplasm from the 3' end. And so the poly A tail is on this 3' end. So it would start digesting it. So the longer the poly A tail, the longer that this mRNA can last in the cytoplasm, which then means that we can have maybe even make multiple polypeptides from that same mRNA as long as it hadn't been digested further. And the GTP cap is important because that is part of the initiation process of translation. And so when we talk about that small and large subunit coming together with the start codon, the GTP cap is an important piece in causing initiation to occur as well. In post-translation, so after we've made the protein, that would be our last way of regulating the gene. And this is really about having proteins that we have proteins or enzymes in our cytoplasm that digest proteins. And one specific one is ubiquitin, which degrades proteins in the cytoplasm. It's essentially so we don't have a buildup of proteins that maybe aren't needed or maybe aren't even being used or maybe we don't want them to be used. And so we have these enzymes that you don't necessarily need to know this process, that will break down the protein or the polypeptide into amino acids. And then we'll reuse these amino acids to make new proteins. And so they are important as well. Lastly, we have one of the dot points here in the syllabus is to talk about how does environmental exposure how does it regulate our gene expression? And so this is a pretty complex topic, but we have lots of things and lots of studies that have been done that show that many things can regulate our genes and epigenetics. And so kind of put all of that here, but the epigenome is what's sitting on top of those genes. So we're talking about those methyl groups and those acetyl groups. And so any of these things can affect your epigenetics. And, something that the epigenome can also be passed down. And so, stress during pregnancy might affect not just you, but maybe your granddaughter. And so because of the epigenome that's being passed. So one example of environmental exposure that causes regulation of genes is in Siamese cats. And so in this cat, you can see here, he has some white fur and black fur, and their fur is what gene is turned on in each one. So the black fur gene is only turned on in places that are cooler on the body. And so this cat the genes for black fur would be turned on in the face and on the ears, maybe on the paws and on the tail, to allow for probably greater absorption of heat. And so the genes being turned on here are sensitive to the temperature of the cat. And so thinking about that, this is a different kind of an example of how the epigenome is affected based on temperature. So we know a lot about the epigenome, still a lot more to learn, but there have been some studies done of twins and looking at, and we were going to look about this more, we'll talk about this more in class, but one example of a study that was done was of Scott Kelly, who's a NASA astronaut, and his identical twin brother, Mark. So Scott Kelly went to space, obviously, and they noticed that there was a pretty big effect on his gene expression of genes that had previously been expressed before he went to space. And so they could really tell the difference in those by comparing him to his identical twin. And so he had a decrease in speed and accuracy. His cognition wasn't affected, but the speed and accuracy was. And they were able to compare him to his brother Mark, and so that allowed for them making some connections between how space travel might affect his gene expression. Lots of other cases of this too, where you see identical twins that might be very, very different, especially if they haven't had the same environmental exposures. We do have some genes that affect are transcription factors, but they actually affect morphology. And so an example of that is the Hox genes. The Hox genes are found in all animals, almost all animals, and they are transcription factor genes. But these transcription factors are causing the layout of the body plan early on in development. And so these genes, while they are present in all animals, they are not in the same place in all animals. And so, for instance, in this fruit fly, this is a Drosophila, these Hox genes are all found on the same chromosome, whereas in animals, in humans and mice, they're found on different chromosomes. But they still lay out the same body plan, and the actual sequence of these genes are almost identical in all animals and cause the same exact thing to happen. So they cause the formation of the thorax and the abdomen and the head. And so during development, and this is partially that these sequences have been conserved through evolution because they actually lay out the plan for the body. And so this is an example of a transcription factor gene that's affecting morphology. Another example of a transcription factor gene is the SRY gene on the Y chromosome. And so we typically would say in biology that a male is assigned male at birth if they are XY, and a female would be assigned female at birth if they were XX. And that is typically true, but in the Y chromosome, there's only one gene that determines maleness, and that gene is called the SRY gene. And that gene is actually a transcription factor for a transcription factor. So it is a transcription factor for other transcription factors, and then those transcription factors go on to the primordial gonad cells and take them down the path of becoming testes instead of ovaries. And so you can kind of think as the default without this gene is female ovaries form. And with this gene, the ovaries, the primordial ovaries would then turn into testes instead. And so one question that comes up then is, moving my screen here, one question that can be brought up in this is it possible for an XX to be male or an XY to be female? And so this is actually possible. When we go back to thinking about meiosis, when we talked about crossing over, very rarely, probably 1 in 20,000, there could be a crossover event between the X and the Y chromosome. And if that crossover event happens to be on the SRY gene, we could end up with an SRY gene on the X chromosome and no SRY gene on the Y chromosome, and then that would be inherited in a sperm, and fertilize a normal egg. And so you could end up with an XX male because that XX male would have the SRY gene on the X chromosome. There might not be any differences, but they are infertile. And hard to know why that might be true. It could be due to Barr bodies or something else that we haven't really discussed in this class. But in the female, that could happen. You could have a female XY if the Y chromosome did not have the SRY gene on it. So just having the chromosome isn't enough. The SRY gene on the chromosome is what allows someone at birth to be assigned male or female and to produce male gonads versus female gonads. So that's about all for this video over gene expression. It was really a kind of a basic overview. We don't go in a lot of detail in this course on this, but hopefully that gives you a good idea that genes it's not just about inheriting genes, but also that the genes have to be regulated. So if you have any questions, let me know.